So, first period said they thought they needed a recording to know what to study for the test, so I'm recording this. The PowerPoint slides are already on Edmodo, and so this is just another recording to let you know what you need to do. So, the things that you need to know about for your test, you need to know about the stages in animal embryology. So, you know that the zygote is the first cell, this combination of the two gametes, and that goes through the 2-cell stage, 4-cell stage, 8-cell stage, and so forth, till you get a solid ball of cells called a morula. As the cells continue to divide, eventually there's not any room for the interior cells to get enough nutrients, etc., so they migrate to the outside and make that hollow ball, the blastula. And over time, as more and more cells are divided, some of those cells start to migrate inside, causing the pushing in of one side of it, forming the gastrula. That process is called gastrulation. Over time, that gives you 2 cell layers, okay, which you have some animals that have 2 cell layers, like the cnidarians. And then over time, a 3rd cell layer develops in between those 2 layers, and you end up with the 3 germ layers, the endoderm, mesoderm, and ectoderm. And eventually, you have the development of a coelom. Now, another thing that you need to know is which phyla of animals have which layers of the germ layers. And if you will look in your animal packet, you can find that information in the tables that we filled in as we took notes. The first couple ones are actually filled in for you. Another thing you need to know is which phyla are protostomes and which are deuterostomes. That's pretty easy, okay? The sponges and cnidaria do not have that, they're not either a protostome nor a deuterostome. And everything else is a protostome except for the echinoderms and the chordates, which are deuterostomes. And remember that the protostome has the mouth forms first, and the deuterostome, the anus forms first before the other opening occurs. On the germ layers, you need to know which organs and tissues develop from which germ layer. So you need to know, like, the ectoderm produces the skin and coverings of things and the nervous system, and the endoderm produces the digestive system and linings of things, and the mesoderm produces the other organ systems like the muscles, as well as the lining of the body cavity and things like that. And again, we discussed those in class. You should have those in your notes. And if you have not listened to the notes recording for that part, then you can go back and listen to that on Edmodo in the folder for Unit 15. On the body cavities, you need to be able to define each type of body cavity formation. That's the acoelomate, the pseudocoelomate, and the true coelomate, and know which phyla are which. One thing that will help you tremendously is looking in that packet and looking at the page that has the notes on the 3 worms, because the 3 different phyla of worms describe the 3 different body plans as far as body cavities go. The flatworms are acoelomates, the roundworms are pseudocoelomates, and the annelids are true coelomates. Everything after the annelids, like the arthropods and the mollusks and the chordates and the echinoderms, are all true coelomates. Everything before the flatworms, that's the cnidaria and the sponges, do not have enough tissues to be called any kind of a coelomate, so they don't even categorize as an acoelomate. Another thing you need to know about is symmetry. The symmetry is whether they're bilateral or radial. Remember that the word bilateral means 2 sides, right side and left side. So if it's an animal that can be divided into right side and left side that are pretty much equivalent to each other, that's bilateral. If it can't, if it's kind of round and has body parts that repeat around the center, then it's going to be radial. Think about like a bicycle tire, how the spokes radiate around the center, and that will let you think about radial. The 2 phyla that are radial are the cnidarians and the echinoderms. The sponges do not have any symmetry. They are asymmetrical, and all other phyla of animals are bilaterally symmetrical. If you look at the general characteristics of each phylum, if you look at that animal family tree worksheet that is in your animal packet, where it divides out the animals by symmetry, germ layers, body cavities, and all those kinds of things, that will give you the ideas of how to figure out the characteristics of each of the phyla of animals. So study that. Know how those are arranged, be able to, you're not going to have to reproduce that on your test, of course, but you do need to know how they fit into that category, into those categories. The chordates, there are 4 characteristics that all chordates share. They have a notochord, which is a supporting rod, like a cartilage rod, or can become bone over time. They have a dorsal hollow nerve cord that develops into a brain and spinal cord. They have pharyngeal pouches or pharyngeal arches, gill arches that can develop into lots of different things like gills or like other kinds of things. And they also have a post-anal tail. Realize also that those four things may not appear in the adult animal. They may see one or more of them may be seen only in the embryo. You need to be able to compare the different types of systems in the different phyla of animals. And if you look at the way the notes tables were arranged, each of those phyla, we talked about how they handled digestion and how they handled their getting rid of their waste products, how they handled circulation and so forth. And that should be able to give you some way to compare the different types of systems in the different phyla. The whole purpose of doing it this way is so that you can see the development of a system from the very simplest animals through the others to the most complex animals, and see how the digestive system, for instance, advanced from just a gastrovascular cavity where digestion occurs and the nutrients are absorbed, to a system that has two openings so that the waste products can go on through and they can continue to eat, to something as complex as a cow and have all the different compartments that do different jobs within the digestive system. You should be able to see that from looking at what happens in the different phyla of animals as we move through the animal kingdom. You also, when we talk about the systems, you need to know which systems work together to do different tasks. And the activity that we did in class on Friday lets you work through how those systems work together. There are a lot of interactions between systems. The whole idea behind this is to show that systems are not isolated from each other. It takes more than one to do the thing, and it's a teamwork kind of thing rather than a bunch of individuals. Explain why certain systems work the way they do, like why does the blood have to go to the kidney to be filtered and things like that. Look through your notes on the systems and see how they kind of fit together. Be able to explain how negative feedback systems work and why negative feedback systems are necessary. This is a really important part for regulating the different systems and the different symptoms or so forth in the various animal groups. Why is it necessary to have a system that will turn something else off when it's present at a certain level? This is really, really important. It's one of the main things that we have to get out of this. Make sure that you understand how it works. Make sure that you're able to answer questions about it, and we should be fine. The test has 40 questions. They're all multiple choice. They're all over embryology and the animals and the body systems. If you will use your packets and use the notes that we took in class or online on Edmodo and look through the worksheets and see how we talked about how things work, you should be fine for this test. If you have any questions, post them on Edmodo. I will be monitoring Edmodo. I will also make an Edmodo quiz that will be up sometime this weekend. So study for your test, ask questions if you have them, and if you are on Edmodo and somebody asks a question that you know the answer to, then you can answer it too. You don't have to wait for me. Study. We'll see you on Monday. Bye.